Why heterogeneous catalysis? Because if you take a surface and you have a molecules going on a surface, you are totally changing its reactivity compared to gas phase. So if I want to go from this compounds to this compound and basically making a reaction, chemical reaction from year to year, if it would be homogeneous, I have to provide some energy to go from year to year. If I go on a surface, the energy pattern is totally different. And very often, chemical reactions on a surface are much easier than on a pure homogeneous system. This is why chemistry is using catalysts all over the time. So this is already a real major conclusion, is that interfaces are a good place for chemistry. Now I ask you, where do we find interfaces or surfaces in the environment? There are many of them. Clouds, aerosols, vegetation, the snowpack or ice pack, the ocean. I'm going to talk about the ocean, but also cities. All these buildings are surfaces on which reactions are taking place, and we totally neglect that. This is in no single air quality model, despite being a real important thing. So we are totally neglecting processes on surfaces, and this on most of the surfaces. So I skip that because you're all aware. PM10, PM25, I don't need to introduce that here, I guess. Am I right? Yeah. So I can just skip all this, which is a kind of, and also this, because we all know. Lyon is also a city where we have air pollution issues. So you can have clear sky and a polluted event, and especially during winter time, we have nicely stable conditions with boundary layer being very stable, and with lots of emissions, especially via biomass burning. So we are during winter time very polluted conditions. Of course, the air we breathe is harmful, it impacts climate, and blah blah blah. Okay, now to my talk. This is a photo of my backyard. It's nice, huh? I typically live here and do my cooking here on the side, and I watch outside. I'm thinking, what is happening there? And this is typically what you would. It's a kind of cartoon we made that you can really find. Maybe I can go already. No. Basically, if you go to the ocean, there is biological activities taking place in the ocean, right? There is life in the ocean. And this activity is producing lots of species, cells. These cells are producing compounds, and these compounds are supposed to degaze into the air, making VOC, volatile organic compound emissions, on the air aloft. Okay. Is this true? I'm challenging this. Why am I challenging this? It's because if you go to the top of the ocean, it's a surface. And we know that the surface of the ocean is enriched with organic compounds and making a kind of thin layer of organic compound which are coating the ocean for most of the places. And this layer is called the surface microlayer. And I'm saying this is making chemistry. And we neglect this chemistry. So this is why we investigated that. Again, interface is a good place for chemistry. And why do we have, why is it a really good place on the ocean? Because we had a lot of surfactant. And the surfactant are present at the interface, potentially packed, or surfactant is something like that, right? Molecules having a polar head and a non-polar tail. It's like a soap. Without surfactant, you could not wash your hands with a soap, would not work. And you have the polar head, which is soluble in water, and the non-polar tail, which is insoluble and goes into the gas phase. And this makes special surfaces. Especially when you go into very concentrated regions like the surface of the ocean, where you have lots of these chemicals around. And now I'm going to present you my view of this interface. It's looking like this. So you have water and lots of stuff on it. Okay? And all this stuff is, maybe I'm just going on the side here. If I just take an example, this is a good example. You have two molecules talking to each other, enjoying being there, exchanging information, basically doing intermolecular interactions. And there are single molecules being very happy being here, enjoying floating around. That's very interesting. But all these molecules are in contact with each other. And for me, this is really a nice place for doing chemistry. Because chemistry in a concentrated environment is typically very efficient. Another point is, in the air, most of the processes are triggered by the sun. So now we have an environment rich in organic, and we have light coming on top of it. And the other point I'm challenging in this talk is, if you go to a textbook, atmospheric science textbook, photochemistry is restricted to the UV region because you need energy to really break bonds, to do photolysis, right? But is that the only thing going around? There's a major reaction taking place in the air, is photolysis of ozone going O∪D, which produces OH radicals, and basically which is cleaning the air. That's very important. But my question is something else happening? So if I take molecule A, which is very famous, absorbing light, the first thing which is happening, it's going into an excited state. Okay? And this is very well known type of Jablonski diagram. You start with a fundamental state, you absorb light, you go somewhere in an excited state, you may do some intersystem crossing, and some molecules, especially organic ones, complex ones, are ending in a triplet state. What happened here? You went from a ground state to an excited state, you gain energy. And I'm always thinking about that when my son who is a teenager is drinking some Red Bull, you cannot stopping anymore, right? It's just run crazy, it's completely crazy. The same is happening with molecules in an excited state. They are going to do chemistry which is impossible in the fundamental state because they have gained energy, and so they have gained strength. And from this excited state, you have lots of possible chemical reactions taking place. Electron transfer, hydrogen transfer, energy transfer, blah, blah, blah. And again, so now we have a situation where we have photosensitizer potentially enriched in this surface microlayer, so on the top of the ocean, and with photon on it. And if this is happening, then you can do chemistry or photochemistry over the full actinic region. And you are not restricted to just the UV, because you don't need to break bonds. You just need to have energy to put molecules into an excited state. So of course comes the next question. Are these photosensitizers really existing? Of course. It's well known for humic acid, for instance. Humic acid, you will find humic acid in every place. It's called different ways. Could be rivers, it could be in rivers, could be in ocean. It's dissolved organic matter, blah, blah, blah. You can find molecules which are more simple, which are produced in the air. You can find molecules produced by combustion processes. You can find molecules coming from biological origins. And these are all known, so we studied the reactivity of all that. I'm not going into details, but all these molecules are reacting extremely fast with various systems. Here you can just take, for instance, the reaction of bromide with this molecule here in an excited state. It's almost diffusion limited. It's as fast as the reaction as with OH. So it's very fast. So this is my first question. There are many photosensitizers around. Another important point, they all carry for most of them, an important functionalities, which is a carbonyl function. I challenge always everyone to find me a surface where there are no carbonyls around. This does not exist. Very simple. Maybe if you go in the stratosphere or elsewhere, maybe. Of course, not all carbonyls are reacting and not all carbonyls are very efficient, but this functionality, chemical functionality, is found in the troposphere almost everywhere. Okay, so now we are here. Ocean, top layer enriched with organics, potentially photosynthesizers. We have light going on it. We make all this strange photophysics going on, and we put this surface into a triplet state. Is this changing the emission of VOCs in the system? So we are stupid chemists, so we do simple experiments. So basically we put an ocean into a beaker, okay, because having a real ocean is a bit expensive. And what we are doing is very simple. We take water and we put these organic compounds in it, which we took as proxy for what you will find in the environment. We took proxy for what could be available as surfactant, an alcohol or an acid, very simple molecule. We took this one because we had them, but also because their properties are known. Okay. And then we do a very simple experiment. We just took a reactor, very small, it's smaller than this, maybe half the size of this bottle of water. We put water, we put a monolayer of surfactant. In the water we put the surfactant. We put light in the system. We flush the system with pure air. And we analyze what is coming out in terms of gases. Okay, this is getting expensive then because we have very advanced tools for doing analytics. And this is a photo how it looks like. It's extremely simple experiment. Okay. So this is now water containing this photosynthesizer for BBA and a monolayer of octanol. So what are we looking here? When we switch on the light in the system, we have production of VOCs. And we do see all these VOCs. Okay? Also this one. So we start for instance with a C8 alcohol and we do C16 products. Okay, why not? Okay. Well, very important information. We also do see products which are unsaturated, containing double bonds. So we start with the molecules being saturated, so having no double bonds, and we do see products having unsaturations. Okay, why not? So we change here the surfactant, we go for the acid. Same story. We do see tens of different products being produced. I'm not entering into naming all these products because it will be by far too complicated. So real complex products being produced. So we investigated a lot of things here. And then we also see if we switch now from synthesizer and go for humic acid, same system. When we switch on the light at a given time in the reactor, we do this for this molecule here. And this is isoprene. So we start from a linear molecules having a carboxylic function being totally saturated, and we produce isoprene. And isoprene is thought to be produced by biological activities only. No. It's not produced just by biologic activities. There is no, in our system, there is no living species. These are pure chemicals that we put in our reactor. And of course, you have to trust me that we are trying to work the cleanest we can. And we do see isoprene. And we also do see isoprene when we go with authentic SML samples. And of course, this is where our problem started. Because this is not possible. How can you go from this linear molecule here to produce this strange doubly unsaturated ramified molecule? This is where I started to have gray area. You can see it here. So this is also quite important. So I'm going over here. Important picture here. So we are using now nonanoic acid. And in blue you have the surface tension of nonanoic acid. So basically the amount, the energy of the surface, but basically the amount of nonanoic acid that you have on the surface. And it goes down and it's leveling off around 1 millimolar. That's classically known. And here is the isoprene concentration we are making in our system. It's basically leveling off approximately at the same concentration. If this would have been a bulk processes, it may have scaled linearly with bulk concentration of the acid, but it scaled with the surface concentration of non-anoic acid. So for us, this is pointing toward a real, authentic surface reaction. So yes, specific chemistry is taking place at the air-sea interface. And I'm coming to that later. These are all data looking at the emission of isoprene in the environment. And basically, if we try to extrapolate what we have in our small surface like that to the ocean, we can explain all the isoprene fluxes being observed on this planet. Of course, it's an overstatement because it's difficult to extrapolate from maybe 50 square centimeters to, I don't know the surface of the ocean, but out of my brain right now. Okay, we do see many products. Okay, I'm just taking isoprene is one minor product, but it's there. But everyone is interested in isoprene. This is why I'm always putting it up front. But what we are producing are other compounds which are major ones, like this one. Alpha unsaturated carbonides, oxo acids of various forms, hydroxylated ones, etc. I don't remember if I put the slide, but this one is very important because it can explain glyoxal formation in the marine boundary layer. We checked all the mechanistic information and we gain information about the chemistry going on. And I'm skipping all that because I just don't want to put too much chemistry, but we can explain, or we have some ideas of what's going on. Okay, so this was our plan. Very simple. We put chemicals, light, we see product, end of the story. Nice. Okay, but reality is always a bit different, right? It was really unexpected that we do see isoprene. Because initially we thought we were looking only at volatile oxygenated products that would just degaze. But no. But of course everyone is telling us, come on, you are stupid, you are using strange chemicals. What is this? This is not realistic, it has no environmental impact. Okay, let's go to authentic samples. Okay, it's working good. Okay, so what we did, we went to people having, okay, Lyon is a nice place, but it has a big problem. If you do marine research, it's that you are far away from the ocean. Okay, but we are very close to many lakes. And people in the building where we were specialized in looking at lake and river biofilms. Biofilm is this greenish stuff that you can find in every stone in a lake or river or whatsoever. So they are collecting those and they are analyzing, they're growing it, analyzing it, blah, blah, blah. And basically they gave us some of these biofilm samples. And we put it in our reactor, it looked like this, and then we put light on this, it looked like this. Okay? And we basically did the same experiment without any addition, right? We just took the biofilm as collected from a lake. Of course, I'm making a big jump here, going from the ocean to non-salty conditions, but again, why not? Let us be a bit open-minded right now. So we are doing the same experiment, same picture, same experiment, same answer. Of course, the details about numbers, exact structure of molecules are different, but you do see here the same kind of picture. When we switch on the light, lots of emission of different compounds. All the the structure are written here. I'm not going to detail them. But basically it looks very similar. And this is now a very interesting plot. This is showing you the traces not for a single compound, but integrated on all the VOCs we observed. Okay, so it's a total flux of volatile organic compound as a function of time for the experiment. And at time 0 we switch on the light. The blue line here is an important one. You see nothing is happening, that's the water. Because we were using living bodies, it was not deionized ultra pure water, it was really groundwater. Okay? Nothing is happening, and in the green line here is the biofilms after 5 days of sampling. The orange one, 6 days, and the red one, where we killed it. And you really see that basically with a dying organism, we do see more and more VOCs being produced. And here, before time 0, it's what this living organism are emitted just by degazing. So this difference from here to here, if I take, of course, maximum because I want my message to be clear, it's a photochemical production. And without light, we do not see VOCs being emitted in the system. You can argue it's a specific system, blah blah blah. I'm happy with this.
So it's the decomposition of this organism which is creating the VOC.
I'm coming to it. Yeah. Okay, so of course this is telling us something is happening when the organisms, these diatoms, these films, these biofilms are dying. Okay. So we spend a lot of time killing them. And we try to do our best to kill them in many different fashions that we could think of, because we wanted to make sure that the way we are killing them has no influence on our observations. Okay? So we kill them by sonicating them, by flash eating them, by stopping feeding them, by removing oxygen, by we are beasts. Okay? So must say. So if you look now at what a biofilm really is, it's cells and with different things connected to them. You have cells, what people call extracellular polymeric substances, which are linked to the cell skin. I'm not a biologist, so I'm just believing what the other people explain to me. And you have at the end also the colloidal fraction, which is a bit further away from the cell. And basically, the biologists are able to make from this biofilm liquids, they're able to make to separate all these fractions. Okay. And so they gave us also cellular fraction, capsular fraction, colloidal fractions to be tested. And this is the same experiment again, and this is exactly what we did. Again, to show the same. I'm mixing now with French. Always the same story. Total VOC fluxes function time. Green is a colloidal, and the blue one, which is the most important one, is a cellular one. What does it mean? It's really what is contained within the cells who are probably triggering the chemistry.
Of dead cells?
Of dead cells. So basically the cells are dying, making cell lysis, opening, releases organic materials, which are acting as surfactant. We saw a decrease of the surface tension for the cellular fraction, goes to the interface and make photochemistry. And this is typically what we are looking at. We also did some analytics on the liquid phase, and we did analytic on the gas phase, as I mentioned before, lots of different compounds being investigated, and because we were not isoprene is so tricky to see isoprene in the system that we did use different type of tools to make sure, also GCMS and whatsoever, and it was always there. So I'm just skipping all this. So what we are suggesting now is that basically you have biology happening in the ocean. After the cell lysis, the products goes to the interface, make photosynthesized reactions, and are emitting VOCs. So what are these VOCs doing? So we can also do chamber experiments where we have our own ocean, and basically put the biofilm in this aquarium there, and put light, put a little bit of ozone, and look what is happening. We're making SOA. Okay, there is no shaking, there is no break, there is no waves whatsoever. We are emitting VOCs, and when we inject So photochemistry was taking place before time zero, and at time zero we inject ozone in the dark. We are making small particles. Then we switch light again and inject a second time ozone. We are making even more particles. So yes, we are making aerosols, which are typically important in terms of cloud formations. And what is really important here is people have seen for a while now, and there are many papers on that. There is always a discussion about, is there a correlation between blooming, VOC emission and aerosols? And some people are saying there is a correlation, there is not a correlation, and some people are saying there is a correlation but with a time delay. And the fact that you do require that you have cell leases explain the observed time delay between the field observations. So we showed that surfactant originating from cells do make very specific chemistry at the air-water interface. These emissions are increasing when death is taking place. It's a new source of oxygenated VOCs. We, for instance, showed that these compounds could explain the observed formation of glycol in the marine boundary layer. And we know that this surfactant are enriched in the SML, which covers something like 70% of Earth. So we are not talking about a small surface right now. And this is what suggests us to say that we have an abiotic production of VOCs, which could explain some of the observation. So typically this is a story. We have activities, biological activities, it makes fatty acid, proteins, amines, lots of stuff. It goes to the surface, it makes photosensitized reaction, make VOCs, make aerosol, and could impact cloud, of course. And what is important here is that, of course, this comes in addition to bubble bursting, etc., because the wind speed conditions are totally different between these two processes. What I'm showing here is really happening at low wind speed where you have stable conditions. While bubble bursting is more efficient when you have waves. And what is important here, these processes require the time to get this material on the surface and could explain the time delay that has been observed between blooming and actual VOC emissions.
A question? Well, that picture would help. It's that biology, you have biology not everywhere, not over the 70% of the...
So this is, there are tools to calculate SML coverage, okay, which take advantage of a satellite picture, blooming events, calculating how much you would get on the surface, and SML. You can get SML maps. So what we did, and this is a paper which has been published in Nature.com this week, or last week? Last week, where we made calculations. So we use these tools for calculating SML coverage. We assumed wind speed conditions, so there is a discussion about up to which wind threshold the SML is stable or not. You can find data or people arguing that it's between 7 or 8 meters per second up to 13 meters per second. I'm not arguing here, so we are just using this data, right, at different thresholds. And we just put the chemistry we observed on top of that. So we can calculate the yield of our products, assuming surface tension and blah, 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 and put yield. And this is, again, I'm just using isoprene here because everyone knows isoprene, but again, isoprene is still a minor product of what we are seeing. And so some people started to have, to look at this. So there was a first cruise going down here, on the south hemisphere, and they say they do not observe anything. But they went in conditions where there was nothing. Another cruise went to the north, somewhere, let me go back to the map, I think they were somewhere here. And they observed something. So currently there are 2 papers out, one saying nothing is happening and we are wrong, but we claim they went to the place where nothing should happen at the time when they were there. So it's not totally an argument against our story. And another paper is suggesting it could be true. Of course, you cannot conclude from there, but at least it gives us some hope that maybe our story is not totally stupid. And this is basically my last slide, and I let you play with it. So basically the outcome is the same in terms of VOC production, but the underlying mechanism is totally different. One other possibility is that you are potentially, you do not necessarily require a collocation of blooming and photochemistry and things like that, so the geographical span could be a bit different. Of course, we do not understand everything what I presented here, because I made a complicated story very simple. Maybe too simple, by the way, or too simplistic, by the way. But it's a crazy idea. Some people are challenging right now, but that's science, right? But maybe the VOCs we are observing in the ocean or in the marine environment do have an indirect biological sources, but a direct chemical source. And again, simply because an interface is a good place for chemistry, we can have really specific processes taking place there.